Here we're going to discuss pyruvate and the pyruvate dehydrogenase complex, which is an important step in carbons entering the TCA cycle as activated acetate or acetyl-CoA. So pyruvate is a molecule that comes from our glycolysis, and it is a 3-carbon compound, and it is going to be transported into the mitochondria. Now, in the mitochondria, we are going to have our outer membrane and an inner membrane. And on the outer membrane, you are going to have a voltage-dependent anion-selected channel, the VDAC, and small hydrophobic molecules will be let in, as well as you're going to have transport of ADP, ATP, pyruvate, which is what we're interested in, as well as other metabolites. Now, the mitochondria, of course, is a bacteria remnant, and so its channels are the beta barrels. And so these are going to enable, on the outer membrane, things to come in. It's a little bit more relaxed than the inner membrane. And then on the inner membrane, you're going to have specific carriers. For us, what we're talking about today is pyruvate. So pyruvate has a mitochondrial pyruvate carrier complex on the inner membrane. So pyruvate will be able to get in and cross both the outer and the inner membrane of the mitochondria. Now, once pyruvate is in the mitochondria matrix, again, this is a 3-carbon compound, and only 2 enzymes can work on pyruvate. The first enzyme is pyruvate carboxylase, which takes the 3-carbon pyruvate and converts it into a 4-carbon oxaloacetate. And so this is using biotin. And the carbon is going to be transferred is from bicarbonate. And then we also have pyruvate dehydrogenase, which is what we're going to discuss here. Pyruvate dehydrogenase is going to take the 3-carbon pyruvate and it's going to generate a 2-carbon acetyl-CoA. And so a carbon is going to be lost to carbon dioxide. And so acetyl-CoA is going to enter the TCA cycle. Now, oxaloacetate here can also enter the TCA cycle, or it can do other things. So this can go to TCA or other. The fate of acetyl-CoA is going to be the TCA cycle. Now, the pyruvate dehydrogenase complex is going to be a very complicated complex. It's going to be made up of 3 different enzymes, enzyme 1, 2, and 3, so that's where the E is. The first enzyme is actually called pyruvate dehydrogenase, and this is going to have specificity for pyruvate. So E1 is going to make pyruvate dehydrogenase complex only work on the substrate pyruvate. And it's a very large complex. It has 30 subunits. And then we have E2, which is a dihydrolipomide sulfur acetyltransferase. And this is going to help to make acetyl-CoA and also have participate in recycling the enzymes so that they can reset and work on new pyruvates. And this has 20 subunits, very large. And then we have a dihydrolipomide dehydrogenase, and this is going to be 12 subunits. Now, one other piece to this is coenzyme A, because we're going to make acetyl-CoA, and so coenzyme A is going to be part of this. So coenzyme A is going to participate in oxidation of pyruvate. We're also going to see this again when we're talking about the synthesis or oxidation of fatty acids. But to make CoA, you're going to take 5 steps, and you're going to need 4 ATP molecules You're also going to need pantothenate and cysteine in order to make acetyl-CoA. So this is coenzyme A, and again you have a pantothenic acid, and you're going to have acetyl-CoA is basically going to be a thiol group. And this is going to react with carboxylic acid, and this is going to form an acyl carrier group. So production of acetyl-CoA, which is going to be our activated acetate, is going to come from pyruvate. And again, this is going to require those three enzymes, enzyme 1, 2, and 3. It's also going to need 5 vitamin-derived coenzymes. So we're going to require our thiamine pyrophosphate. We're going to require our FAD, our flavin adenine dinucleotide. We're going to need NAD, which is nicotinamide adenine dinucleotide. Lipoate is also going to be needed, and then of course CoA, which we just talked about, which has that nice sulfur group. All of these are going to be required to take pyruvate, which is a 3-carbon, to acetyl-CoA, which is a 2-carbon. And again, you're going to release 1 carbon as carbon dioxide. Now these are the 5 coenzymes. Again, they're going to come from vitamins, so we need vitamin B1, riboflavin, pantothenate. We're going to need vitamin niacin and lipoic acid. Now the other thing that this shows is this blue color. So thiamine, pyrophosphate, our FAD, and lipoate are going to be tightly bound to the coenzymes. Our coenzyme A and our NAD are not going to be tightly bound, so they're going to be able to come in and leave. So the 3 enzymes are E1 and E2. E1 is going to be bound by our thiamine pyrophosphate. E2 is going to have lipoate, and E3 is going to have FAD bound. Now this list does not include our coenzyme A and our NAD because they are not tightly bound to the enzymes. In addition, you can see these aggregates under an electron microscope, and so these are going to be the Pyruvate Dehydrogenase Complex because there's multiple copies of these enzymes in our mitochondria. So here's a diagram of the PDH, and you can see that we have here pyruvate dehydrogenases E1. This is going to have the specificity for pyruvate. Pyruvate is going to lose 1 carbon in the form of carbon dioxide and is going to transfer the other 2 carbons to thiamine pyrophosphate. Now thiamine pyrophosphate is going to transfer the 2 carbons to a sulfur group of our lipolysine. And here you have our sulfur group attached to the 2 carbons. Enzyme 2, which is dihydrolipotransacetylase, is going to transfer those 2 carbons to our coenzyme A, to that sulfur group. So you're basically going to replace this hydrogen with our 2 carbons to generate acetyl-CoA. Now acetyl-CoA is going to go off and go to the TCA cycle, and the rest of the enzyme is to recycle or reset all of the different coenzymes so that you can make another acetyl-CoA. So in this case, you're going to have reduced lipolysine, and so you're going to donate your electrons to FAD to generate FADH2, and then you're going to recycle your oxidized lipolysine. So you're going to go remove those hydrogens, transfer them to FAD, and have a oxidized form. FAD is going to be tightly bound to enzyme 3, dehydrogenase. And so in order to get the access to those hydrogens and also to regenerate the oxidized form of FAD, you're going to grab those hydrogens by NAD and generate NADH. You're going to lose one of those hydrogens in that process. So in this step, step 1 is the rate limiting step. And again, carbon dioxide from the carbon 1 is going to be reduced. And then carbon 2 is going to attach itself to the thiamine pyrophosphate. Now step 2 is also carried out by the pyruvate dehydrogenase, and this is going to have your electrons are going to be reduced on the sulfur groups of your oxidized lipolysine, and you're going to get the reduction on that sulfur group. So one of them are going to take the hydrogen, the other is going to take the 2 carbons that were donated from pyruvate. And then step 3 is going to be carried out fully by enzyme 2, and this is where you get the oxidization and the formation of your acetate or your acetyl-CoA activated acetate. And again, you're going to transfer this hydrogen, now goes on to one of the other sulfur groups, and then the 2 carbons are donated to your coenzyme A. And then finally, 4 and 5 is to reset your lipolysine to convert it from reduced to oxidized, transfer those hydrogens to FAD, and then pass those hydrogens on to NADH. So in summary, pyruvate, which is a product of glycolysis, will need to be converted to acetyl-CoA. This happens in the mitochondria matrix. The pyruvate dehydrogenase complex is very complicated. It has 3 enzymes. Enzyme 1, which is going to have the specificity for pyruvate. Enzyme 2, which is going to transfer your carbons to coenzyme A and generate activated acetate or acetyl-CoA. And then enzyme 3, which is going to capture the hydrogens and pass them on to eventually NAD. So NAD can also go to the TCA cycle.